Okay, I'm Yutanouchi. I'm a graduate student in Linchon's lab from Duke University. So today I'm going to talk about, well, my title is A Synthetic Biology Approach to Understanding Bacterial Program Cell Death and Implications for Antibiotic Treatment. So a little bit of history. So when I joined Duke as a graduate student, this is what I thought bacteria are. Like, you know, it's a single-cell organism. It's very small. You need a microscope to see it, and it just keeps dividing, and very blunt life like this. But it turns out to be, as everybody knows, it's not true. And now, even starting from a single cell, as it divides and multiplies, it diverges into very distinct phenotype, and could be explained by biostability that Jeff Gorgeous. Well, it's population level, but biostability or just stochastic noise in gene expression. So it's more like this. It's a colorful picture. And as we all know now, that that's not the end of the story. They actually are very, very social. So they engage in different social behaviors. Of course, they don't form Beatles pattern, but they form or they perform different population level behavior. And as we already heard in this morning, they do sense their density through quorum sensing, and they decided to when to act at the population level. And they also form biofilm, which we're going to be hearing about after my talk. And they also do collective swarming. And so I think understanding these social behaviors is very, very critical. It's at least as critical as understanding our own body, because we have these, it's said that 10 times more bacterial cells in and on our human body. So and in terms of number of genes, it's probably 100 times more than our own gene sets. And it's once said that by Bonnie Bassler, you think of yourself as human beings, but I think, or she thinks of us as 99% bacterial. So I think it's probably 100 more times important to understand microbes or social behaviors of microbes than understanding our own cells. But don't blame me for that. So it's like bacteria having fun, engaging in different kind of social behaviors in and on our body. So let me emphasize again, it's very important to understand what they do in our body. So today, I'm going to specifically talk about bacterial program cell death, which is one of the social behaviors that bacteria engage in. And I studied this using E. coli cells. And first, let me explain what do I really mean by bacterial program cell death. So here, what I simply mean that there's some genetic death program that responds to environmental stimulus or stress, and it executes its death program to cause cell death in bacteria. So if you look at the population, this is what typically happens. You have a population of cells, and it receives stress and performs this program of cell death. And some of the fractions, some of the cells end up dying, and then that's what happens. So the question here, the central question here is that why do they even do that, right? Because they're not multicellular organisms like us. It's single cell. So once they die, it's all over. So it's like, if I die, I'm gone. So the central question is, why do they do this? And if you look at the nature, natural systems, there are quite a few examples of programmed cell death. And a common explanation of why they do this behavior is that death is actually altruistic, and it represents ultimate form of cooperation. So on the left, I'm showing sort of a general framework. Again, you have population of cell, and upon stress, some of the cells in the population die, but instead they release this public good. And this public good, in turn, counteracts the stress and alleviates the stress, and then in the end, it provides overall benefit to the population. So it's an altruistic behavior. And I'm listing a few examples here, like Streptococcus pneumoniae. When they try to invade us, when they enter some host environment, they initiate this autolysis and release the the variance factor called pneumolysine, which is very critical in their success in invasion. And there are more examples like this. But the specific question I'm asking in this study is that, first, is dying really better than not dying? And if so, under what conditions does it become beneficial to the population? So to answer this question, or address this question, I take a synthetic biology approach. So if you go to syntheticbiology.org, they have some definition of synthetic biology. You can argue for, argue against this. But they say synthetic biology is the design and the construction of new biological parts, devices, and systems, and the redesign of existing natural biological systems for useful purposes. So the key point is this useful purposes. So if you think about useful purposes of engineering bacteria, microbe, maybe you typically think of like some chemical synthesis, biofuel, cosmetics, polymers, or maybe some therapeutic application like cancer-killing bacteria or re-engineering of microbiome, re-engineering of immune cells. But to me, the useful purpose here of synthetic biology is to use synthetically engineered system as a model system to study certain biological phenomenon. So when you usually study some natural phenomenon, you may take mathematical approach. So you build a mathematical model to describe the system, and then maybe you can do different things. But it's sort of, I'm putting two scales here. One is controllability and complexity/reality. So of course, mathematical model, it's what you build and what you run on a computer. So you have pretty much full controllability on your system. So you can change whatever parameter you like, stuff like that. But on the other hand, it's a model. It's a hypothetical thing. So it's very far from, most likely far from reality, or it may not even entail the full complexity of the system. On the other hand, you can study the system as it is in the context of its nature. So there you have, it's your reality. It's a reality of the system because you study the system as it is. But you may not have as much controllability as you'd have with mathematical model. So the idea is to, OK, let me construct a synthetic system to mimic the phenomenon of interest to study so that we can have some sort of middle ground. So you have, it's still biological systems, but you have, so it's closer to the natural system, and at the same time it has a great degree of controllability. So again, to reiterate, so synthetic system is an actual living system. So it's still within a biological constraint. So it's still bound by that limit. And at the same time, it's a well-defined system because you engineer it so you know what you did to the system. So it's very well-defined. And also, since you know what you put in, you can draw a clear mapping between what you do to the system and what comes out from the system as a result of it. And again, since you can choose what to put in, you can make your system very controllable. So if you want to change, if you know certain parameters that you want to change, you can construct a system in such a way that you can control that parameter experimentally. Okay? So again, coming back to program cell death, or now I call it program altruistic death, or PAD. So I constructed this system in E. coli to sort of capture the basic logic of program altruistic death. So, and the system consists of two modules, suicide module and public good module. So suicide module is a module in response to this stress or antibiotic, 6-APA. It induces this lysis, E-lysis gene and then causes cell lysis. So in response to antibiotic, it lyses the cell. So it's suicide, hence. And the other one, the public module, is just a module expressing this gene called BLAM, which is a modified form of beta-lactamase that can degrade 6-APA once it gets outside under a controllable promoter. But this BLAM has to be released by cell lysis to degrade 6-APA. So this is a full full circuit. So I first constructed these modules and experimentally verify the function of each module. The first is suicide module. So here I'm showing time-lapse movies of cells with, green with suicide module, red without suicide module. So an agar contains that antibiotic 6-APA. So the expectation is that green cells will lyse in response to this 6-APA, but red cells don't. So that's what I saw. So over time, you see that the number of green cells decreasing. And I quantified it, and it was consistent with this movie. So for green cells, I had probably 60% of lysis after 3 hours. But for red cells, it's probably a little less than 10% lysis after 3 hours. The reason for this is that the 6-APA itself is weakly lytic, so it lyses the cell a little bit. But lysis was greatly enhanced by this suicide module. So it's good that the suicide module is now working. And the next one is the public module. So I did a couple of experiments to prove this point, but I'm just telling you the conclusion. So BLIME, again, it's a modified form of beta-lactamase, which actually sits in a cytoplasmic as opposed to periplasmic space. Wild-type BLIME sits in periplasmic space. So what happens is that when I add 6-APA, 6-APA binds to its target, which sits in the periplasmic space. So BLIME doesn't have an access to that 6-APA, so it cannot protect cells from the action of 6-APA. So when it's inside, there's no protection. But when other cells lyse and release this BLIME, now this released BLIME in the extracellular space have an access to 6-APA, and it can degrade the drug from the outside. So it can reduce the concentration of 6-APA and sort of therefore help or protect the survivors from this drug. So with this system in my hand, I first tried to see if this PAT cell can actually grow better than this control cell, or no program death cell, or NPD, in a separate culture. So my experiment is going to be all separate. So I built simple model. And here, both cells growing to this point, and then I add 6-APA to both media. And the PAT strain exhibits drastic lysis because of the suicide module, but later it can recover very quickly because it can degrade 6-APA very, very efficiently. On the other hand, the control strain, NPD strain, cannot lyse as effective as PAT, and hence there'll be less BLIME or public good outside. So they cannot degrade 6-APA as efficient as PAT strain does. So it sort of tolerates the stress, and it grows very slowly. And then I did experiment, and I saw exactly like that. So the red, again, is a PAT strain. I saw drastic cell lysis upon addition of 6-APA. And it took probably like 18 hours to start recovering. But PAT strain sort of, it lyse a little bit. Again, 6-APA itself is a little weakly lytic, but it sort of grew very slowly. And there's a critical time point above which PAT actually exceeds NPD growth. So this is good. So in principle, this scheme of altruistic death can pay off at the population level. So if you compare monoculture, there's a condition. Under this condition, dying is worth doing, or it's worth dying. Yes. Right. I mean, if they all die, then nobody's gone. I'll actually get to that, but I don't control the stochasticity, but I control how much this E gene is expressed. But I'll get there. So that's exactly sort of the agenda here. So as I described earlier, a nice thing about this system is that I can control critical parameters in a very controllable manner. So I decided to control 3 different things. The first is the degree of public good production, which determines how much benefit per cell death is gained by the population. And the second is, as it was just pointed out, E gene production rate or death rate. And the last thing is the degree of stress or 6-AP concentration that sets sort of the degree of stress that cells have to counter. So let me first look at this public good production. So as I explained earlier, this BLIME induction, BLIME expression is simply controlled by IPTG. So I can vary IPTG concentration to modulate how much BLIME is released per cell death. So the modeling suggests, and then very intuitively, you have to have high enough public good production so that the PAT strategy or death strategy becomes overall beneficial. So there's a critical threshold here, and experimentally, that's what I observed. So on x-axis, I'm changing IPTG concentration to modulate BLIME expression, and on y-axis, I'm showing the final cell density, 24-hour final time point of each cultures. So above here, PAT strain actually overtook the NPD strain. So it's consistent with the model. So you need to have, indeed, high enough public good production for this strategy to be beneficial. OK, the second, this is I think what you asked. So second is E gene production rate which determines the degree of death. So let me first go through some intuition. So again, this is the conceptual framework. You have cells and this partial lysis, release of public good, and that reduces stress. So what if you decrease the death? So now you have less death, but you also have less public good, right? So the arm inhibiting stress is now weaker. So though you start from sort of higher density due to less death, you also have less power to counteract this stress. What if you increase death? So now you have more public production resulting from more death, and this arm is now stronger. But you start from lower density after the stress to recover. So you can imagine that there must be some optimal degree of death that maximizes the growth of its population. Okay? So to test this notion, what I did was I wanted to control the expression level of this E gene. So what I decided to do, unlike public good production, I can't just add some chemical to control it. But what I could do was to change the ribosomal binding site, which determines the translation of E gene. So there's a nice algorithm published a little earlier that you can actually design RBS strength of your gene of interest to control translation rate. So, and this shouldn't change the transcription profile of this promoter. It only changes translation rate. Okay. So I designed. Sorry, it's kind of. I don't want to. So I designed 4 different RBSs to modulate E gene. So I call them iPad 1 through iPad 4, which stands for intermediate program altruistic death. So when I did this, there was only iPad 2 or even iPad 1. So I thought I was going ahead. But now it's unfortunate that Apple also has iPad 4. I should have patented this. But anyways, so I did this. And first I characterized these variants. So now here, on x-axis, I'm changing different doses of 6-APA. And on y-axis, I'm just plotting cell density after 3 hours of 6-APA treatment. So that mirrors how much lysis has happened. Okay, so the blue one is the NPD strain, which doesn't have any E gene, lysis gene, and the orange one it's the original PAT strain. So IPAT 1 through IPAT 4 nicely sit in the middle. So I now have a gradient of lysis, degree of lysis. Okay, so now I first ran a simulation to see if I can actually find this optimal degree of lysis. So again, on x-axis, I have death rate in the model, and y-axis is some cell density after 24 hours. So indeed, I saw this. It's not as sharp as I want it to be, but there is an optimal cell lysis or death rate. And as I change public reproduction, as I did earlier, it may become sharper. And if I go below a certain threshold, it actually disappears. And no death is the actual optimal strategy. So there's a shift in optimal death rate.
Decreasing public good expression?
Decreasing, so this direction, I'm decreasing public good production, yeah. So I verified this experimentally. So with the blue line, it has highest blind expression. So you have optimal probably here. And as I decrease the expression of public good, the optimality shifts to the left, and then in the end, it disappears and it becomes just monotonic. So it's consistent with the model and intuition, and I found optimal death rate in the system. And lastly, I changed 6-APA concentration to see what happens. And intuitively, again, since 6-APA is it's an antibiotic or stress, intuitively, if you increase stress, you'd expect the final growth to get smaller and smaller, right? So overall growth should depend monotonically on the stress level, which turned out to be the case for low death case. So the control strain and a strain with lower death rate, it turned out to be the case. So I got this monotonically decreasing function as a function of 6-APA. But when death is high, there's something interesting happening. So when death is high, I could get this non-monotonic behavior. So it initially drops, but there's some sort of maxima at some level of 6-APA. And of course, if I keep increasing 6-APA, it would go down. But the point is, with high death rate, I could get this non-monotonic behavior. So I wanted to test this. And again, I did the experiment. And that's what happened. So these 3 lines represent the strains with relatively low death rate. So the control strain and IPAT 1 and 2. And the other 3 curves represent relatively high death rate. So as you can see clearly from this orange line, you can have this bump here, which is consistent. Of course, it doesn't look as good as this, but it still, in principle, shows that with high death rate, this non-monotonic antibiotic response can arise.
Just what is it that you're really probing? Because I mean, this is somehow the saturating cell density, right?
No, it's still increasing.
Okay, so
at the time point that I took.
Okay, so they would. I mean.
No, it's not saturating. So it's not. Well, if you look at the time course, it's still increasing slightly.
Okay, but it wouldn't reach the
no,
high density that it gets.
I don't think so.
Right. So it's somehow, I guess this always happens. You get antibiotics and it decreases the final density.
Right, and also I killed the cell, right? So
Sure.
the nutrient level wouldn't go back to the same level as the initial. So.
Are you sure they deal with the same cell, that they are not evolved?
So So what I did was, after 24 hours, I sampled like 100 colonies, and actually 50 colonies, and regrow them and see if they behave the same. and most of them behave the same. So I don't think whatever I observed here is due to mutants or anything. They're still intact, at least functionally. I didn't sequence like genome, but I sequenced.
The fact that they behave the same is not evidence that they're the same cell. They may
Right.
change genotype, and it may be the reason, I think. They may evolve something, because they are under
Right.
stress.
But
And, bacteria, they evolve very quickly if they're in this condition.
right. But I did this many times, and they're all consistent. And I don't know how likely it is for them to evolve in the same way.
Oh,
Every
it may
time.
be different way, but the same effect. They increase. They
Well-
try to
Sure.
survive. Therefore, they should evolve something. They may be different.
Yeah, sure. But it's still consistent with what I think it is. So yeah, of course I can always go back and sequence them and find some mutation,
I would
maybe.
advise.
But
Oh.
he showed it's not heritable. He showed it's not heritable, right? Because you
Right.
grow it non-selectively and we do this kind of see the
Well, not the same experiment, but they still maintain the lysis capability. So because that's most that's
surviving ones can still be killed. That's
yeah that can still be killed because the most obvious mutation is that they lose this function and not lysable, but they were still lysable, or yeah. I don't know if that term exists. Right, they're lysogenic, yeah, in some way. Yeah. So that's as far as I did. Okay. And coming back to this non-monotonic behavior. So I look into some literature and see if this has ever observed. And indeed, this non-monotonic antibiotic dose response was or had been observed much, much earlier than what I did the experiment. So it was first described in a paper published in 1948 and was found by this Dr. Eagle. So later people call this Eagle effect. It has nothing to do with the bird. So Eagle effect. And later, so in this experiment, he used different strains of Streptococcus variants and look at the response to penicillin. So on x-axis, it's a penicillin concentration, and y-axis is the final cell density. And they actually observe very, very drastic non-monotonic response to this drug in some of the strains. And later, different groups found a similar phenomena in not only beta-lactams, but quinolones or aminoglycosides antibiotics, and not only in this Streptococcus strain, but different species of bacteria. And there have been a few mechanisms that have been suggested. One is, which is kind of obvious, it's self-antagonism of the drug. So when the dose of the drug becomes higher, the drug itself becomes ineffective. So that could be one reason. And the other is that some resistant mechanism against that drug is induced only at the high level of drug. So that's another possible explanation. And here, what I found is it's slightly different. So it's a release of some enzyme. Oh, okay, 5 minutes. By cell death, and that can counteract this. So it appears to be. Sorry. Yeah. So it's slightly different from those mechanisms that we proposed, and implications are that for the same antibiotic, maybe more sensitive strain thrive better or survive better. And for the same pathogen, a more potent antibiotic or more lytic antibiotic may be actually less effective. And finally, the treatment may appear effective initially because they actually lyse, and you see reduction in cell density. But later, they may come back, and it appears to relapse later. But it could be mediated by this cell death mediated release of public good.
And your experiments are very convincing that this is what's going on for your
Right.
strains. But they don't prove that that's what's happening for any of these
Right.
other- I mean, have you actually seen it in any of your strains, any normal strains in the lab, not engineered strains?
So what I had was, so So this is with modified beta-lactamase, right? But I swapped that with wild-type beta-lactamase, and it still happens because
With 6-APA.
with 6-APA.
It's still your engineered strain, though, is that right?
Right.
I guess it's just that we have not seen the eagle effect with E. coli and any of the beta-lactams that we've
Right,
played
right.
with. And I'm just curious whether, I believe that it exists or something.
Right, right, right.
Because it would be interesting to know if that
Right.
is
Well,
a mechanism.
at least there's no known report of this type of ego effect.
From that eagle or whatever, no. I don't know whether you can get it. Just get a strain, right? Test whether they actually release toxins. Release this.
Beta-lactamase.
No, release.
Something.
Even go through cell death.
Right.
Program cell death. But I don't know whether they can get the strains. Like whatever that strain was.
The streptococcus. Yeah. I'm pretty sure that's some clinical isolates or pathogenic strain. Yeah, that's an interesting thing to try out. And if you have a whole bunch of strains, you can test whether that shows ego effect under some condition or not. Yeah. So I think I'm running out of time. But in summary, so I created this nice toy system that represents the basic logic of program altruistic death. And I show that the population level advantage is dependent on public reproduction, and there exists an optimal death rate, and the ego effect may arise because of the presence of this altruistic death. With this, I want to acknowledge my PI, Lin Chun Yu, and colleague Anand Pai, collaborator Nick Bushler, and other ULab and former ULab members, and also, of course, funding. Thank you very much. Yes.
This is a very nice mechanism, and I think it may provide an alternative mechanism to explain bacterial persistence in antibiotic treatment. But now I wonder, this optimal death rate, this is a combination, it's a balance between your expression of the suicide module and this release of the beta-lactamase, right? So that this optimal death rate actually depends on, for example, I have both of those genes are expressed at a very high level, or compared to another set of cells that I have both of genes that's expressed at relatively low levels, but they all can, counterbalance with each other. So under what type of conditions that the cell actually would have the same, would have the same optimal growth rate for the whole population? Do they have given the exactly same effect, or does it matter? Or as long as you have a good combination that you balance the release of your enzyme, the lysis of your cell, then you would achieve the same population fitness.
So are you asking whether I can get the same population density with different combinations of?
Yes, over those two genes, over those two modules.
Yeah, that's so. For example, like here, like they're all like collapsing. So, or maybe this one and this one is very close.
So this means that you want, for example, have this. You want the cell. So you want both of them to be at a very high level, and then that would favor to a more optimal population growth.
So I'm not quite understanding the question.
I'm just wondering if you have two different types of cells,
Mhm.
and both have both have those two modules.
Right.
And one set of cells have very high expression of the suicide module and also this beta-lactamase
Mhm.
module, and another type of cell have an expression level for both of them are very, very low.
Right, right.
Do they give you exactly the same population fitness?
It could, yeah, yeah. So for example,
It doesn't matter what's the exact expression level.
in terms of monoculture population fitness, it shouldn't matter. But maybe if you mix different strains, that would matter, like how much lysis is happening and how much beta-lactam. Of course, if you lyse more and release more publicly, you may be more prone to cheating. But I didn't study mixture dynamics in this study, so that's something interesting.
Supplemental figure, didn't you?
Yes, I did.
One of the referees might have asked for that.
Yes. That's just to show that this death is indeed altruistic. So if you mix like control strain and path strain actually, or control strain takes advantage of path strain, so the frequency changes over time. So that's just to show that this is indeed a social behavior or public good.